All right, we're starting a little bit late because I carried on the lecture a little bit late, and I do need 15 minutes to convert the lecture before I start another lesson. So that's why we were starting late. Although I actually only needed about 1 extra minute. Let's see. Let's go here and take a look at what we're doing. So if you look, this week we're only doing Lab 4 on stains. And then, which is really today, on Wednesday, we're just going to be catching up and doing a little bit of a review. I thought about moving Lab 5 down and moving Lab 6 down. I decided not to do that. However, I might change the labs, and that is, you're going to be needing a lot of time to work on your unknowns project. And so I might move Lab 9 down to Lab 8. So I'm telling you there might be a change in the schedule, and then we will just not do Lab 8. Any questions about what we're doing today? We're doing Lab 4 on stains. All right, no question. Am I recording this? Yes. Share the screen. So let's go over Lab 4. Just a minute. I'm trying to get there. Am I too far? Yes, I am. So when you're doing Lab 4, please note that there's a worksheet to Lab 4, and do your worksheet from this link. And you don't have to print it out, but you should save it down and then put your changes in it, and then upgrade this file. Somebody uploaded the whole lab file, and I really don't want that because it's a much bigger file. You know, this isn't going to work here. Let me see if I've got it elsewhere. Might not be down here yet. Nope.
You can expand it right from that one page in Canvas.
Yeah, I know, but oops, lost it. Got too many things here, and there we go. That's why I couldn't find it. All right, so do the reading in the textbook, and there's a lot of it for this one, but it's mostly just on the stains. There's going to be some video clips and a virtual lab. Upon completing this laboratory, you should be able to do these primary learning objectives, and I'll have you read that. Because of their extreme size, bacteria are transparent, and when you look at them under the microscope, let me see if I can put that over here. No, that's not going to be good either. Let's go there. The bacteria will be small and transparent under the microscope, and so you really need to use a stain. All the stain does is increase the contrast between a cell and the background of a slide. Or, well, actually, yeah, it does do that. And that's by either, if it's a basic dye, it stains the cell, and then the background is light. Or if it's a negative stain, it stains the background and then the cell is light. Either way, it increases the contrast between the cells and the background. And then you have a much easier time viewing your specimen under the microscope. Before you work, somebody's got their microphone on. Can you mute that, please? I'm not finding anyone at the moment. All right. Before you look at a specimen under the microscope, you have to make a bacterial smear for looking at your specimens. And a bacterial smear will be a very thin layer of cells. Ideally, it's only one layer of cells so that you can see the cells very well. And then also, because it's one layer thick, it's very easy for the dye to get in, and if it needs to be washed out, it'll be easy for the dye to wash out. As we mentioned on the gram stain, if you have a multiple layer of cell, it's hard for the dye to get all the way through the layers of cells to the lowest layer of cell. And then if the dye is supposed to be washing off, it's hard for it to wash out of all the layers of cells. So ideally, the bacterial smear will be very thin and only one layer of cells thick. And how you obtain that is by obtaining a clean slide and then adding your bacteria just a little bit, and then using the loop to, I'm trying to show that, to spread the bacteria over the slide. And then allow the smear to air dry and then heat fix. Why we do the air drying and the heat fixing is that otherwise when we go to wash the slide, like in the staining process, if it isn't air dried and heat fixed, all the bacteria will just run off the slide. And if you air dry and heat fix, that will attach the bacteria to the glass of the slide, and then when we're washing, the bacteria don't run off. The advantage for also air drying and heat fixing is that actually kills the majority of species. It won't kill an endospore, it won't kill mycobacterium, but for the majority of the species, air drying and heat fixing will kill the slide. Okay? And the heat fixing is very light. When you're holding the slide, try and hold it here. So pretend this is the slide. You're exposing it to the heat here. If you can see where my hand is. And you're holding the slide over here. All you do is you wait until the glass is slightly warm at your fingers. You don't want it hot. You just notice a slight elevation in the temperature. And that's how you heat fix. Excuse me, just sneezed here. You use many of the common dyes for staining the cell. Angeline, can you mute your, there you go. All right, so let's talk about simple staining. A simple staining is when you're staining a bacterial smear with one dye. Usually it's a basic dye, but it doesn't have to be a basic dye. It could be an acidic dye, and that would be a negative stain. And a basic dye will stain the bacterial cells, and it will stain everything one color. So if you had a mixed population of cells on the slide, there would be no difference in the stain. Like they would all be, if you're staining with methylene blue, all of the cells would be staining blue. A differential stain is where you're using two or more dyes to show differences between different cells. And the most common differential stain is the gram stain, and it's looking at differences in the cell wall composition. And the gram-positive cells have a thick cell wall with a thick peptidoglycan layer. The gram-negative cells have a thin cell wall with a thin peptidoglycan layer. All right, so those are the differences between the simple stain and the differential stain. Differential stain uses 2 or more dyes, and it stains the cells differently, so the 2 different cells will look differently. There are also a number of special stains, and we use them to stain particular structures in bacteria or in microorganisms. And you could use a special stain to stain the capsule, a special stain to stain flagella, perhaps to stain endospores, although there's also a differential stain for staining endospores. All right, any question about the 3 different stains? Simple stain, differential stain, and then lastly a special stain. So on a simple stain, they will all look the same, but you might be able to tell differences in the shapes or in the growth arrangements. You can take a look at the shape of cells, and there will obviously be cocci, bacilli, and let me see if I got this spirilla. You can also look at the growth arrangement, although to see the cellular growth arrangement, how the cells are growing together, you really need to grow the cells in a liquid culture and then make your slide from the liquid culture. When you make the cells from a plate or a solid culture, the cells tend to be sticking together, and then when you put them onto the slide, they'll stick together, and you might not see the real growth arrangement unless you're growing them in a liquid culture. Any question about that? Anyways, for Streptococci, you can see individual cocci, diplococci, Streptococci, cocci growing in tetrads, cocci growing in Saxernia, meaning 8 cells, Staphylococci, Sarxenia, that's the word I was looking for. Bacilli, you can see single bacilli, you can see bacilli growing in diplobacilli, you can see streptobacilli. You can also see them, instead of growing length to length, meaning a sausage length for the bacilli, you could also see them growing in palisades, and that is where they are stuck together on the long side of the cell. You could look and notice whether your culture was pleomorphic. That means they have more than one shape, and that's not shown too well here. And then the spirals. The spirals are unique, and that is they usually only have spirilla that are singlets. So the spirilla don't tend to grow together, they're usually just singlets. And you can't have bacilli that are singlets as well as cocci that are singlets. Now that you've learned the cellular growth arrangements, let me warn you that when you look at a cellular growth arrangement, you'll never see only one. So if you have streptococci, if it breaks, there will be diplococci. And if the diplococci breaks, there will be single cocci. So you'll never see just one cellular growth arrangement. The growth arrangement will be many. And what microbiologists do is when they see multiples like singlet cocci, diplococci, and streptococci, they go on the highest one. So they'll say it's a streptococci. Any questions there? We talked about the gram stain briefly. Let's mention that it was invented by Hans Christian Gram, and he devised it to differentiate between a patient's cells and a bacterial infection, a microbiologist started using it and they said, my gosh, this gram stain separates out gram positive and gram negative. And it's the most widely used stain in microbiology because it can separate cells on the basis of their cell wall. Any questions about that? I won't ask you on what year he invented it, but you really should, when you're using the gram stain, start it with a capital G because it's named after somebody. Okay? Any questions about that? He uses crystal violet as the primary dye, and if bacteria retain that dye, we call them gram positive. Bacteria that do not retain the crystal violet, we call them gram negative, and then they will appear pink because they are colored by safranin, which is the counter stain. And here we're looking at a mixed culture of cells, and we see gram positive bacilli, gram negative spirillum, gram positive bacillus, and maybe gram positive cocci right there. And I mentioned the gram positive reaction is based upon the thickness of the peptidoglycan layer. So when we're looking at this is kind of hard to see. Let me move that out to the side there. Oops. The gram-positive cell looks like this, where there's a thick peptidoglycan layer, and I'm not entirely sure what those are. I'm going to guess that's the, what is that called? The molecules running through the peptidoglycan and the gram-positive cells, and what's the name of that molecule? Nobody else remembers. I'm drawing a blank on that. I'm sorry about that. I'll have to look it up. And that's different from the gram-negative cells. They do have a peptidoglycan layer, and please don't tell me it's one layer. It's just a thinner layer. If you look at that, you can sort of see the peptidoglycan layers running throughout it. And gosh, I would say that's at least 30 layers. But it's much thinner than this gram-positive cell, which, I don't know, maybe 100 layers, maybe more. It's hard to tell. And the gram-negative cells also have a outer membrane, which is lipid-rich. The lower one, it's only phospholipids and proteins running through it. The outer one, it does have phospholipid in it, but it also has lipid A, which is an endotoxin that's in it. And that would probably be this one here, and that one there, and that one there. And the lipid A is connected to a saccharide, so we call that a lipopolysaccharide. Any question about that? All right, so you first stain the cells with crystal violet. That's the primary stain, and all the cells will stain purple. You then add grams iodine, which that is a stain, but we're not using it as a stain. We're using it as a mordant. The iodine complexes with the crystal violet, making a larger molecule than the crystal violet alone. And then that crystal violet iodine complex for the gram-positive cells will not wash out in our next step, the alcohol acetone decolorizing step. And you apply this briefly to remove the excess crystal violet. And the best way for doing it, if you're looking at your slide, have I mentioned this? I don't remember, is you add the decolorizing agent, and when the slide stops running with the blue color, you stop adding the decolorizing agent, and it's about 5 seconds. And then you immediately water rinse to get the decolorizing agent off the slide, because If it's still on the cells, even a tiny bit, we'll continue pulling out the crystal violet iodine complex from the gram-positive cells, and we don't want that. So you immediately water, rinse, remove the water, and then you stain with safranin, which is the counter stain, and it will stain any of the cells which are clear, which hopefully are the gram-negative cells, and then we'll stain them pinkish color. Okay? Now, with that stated, some of the bacteria do not stain well with the gram stain, and we call them gram variable. It's mainly bacilli, particularly of the family Mycobacterium. And so here's the stain that was done on these cells. And as you can see, some of them are staining gram negative, some are staining gram positive. Does anyone notice a difference? Like, can you tell me that cell there? Do you see a difference between that cell there and this cell here? Or that cell there? And like this cell here, or that cell there? Did you notice a difference there? Come on, somebody's got to notice a difference.
Are you referring to how some of them appear kind of both pink and purple at the same time?
Yeah, that's one difference.
Yeah, that's one.
Can somebody else state?
Can somebody else state?
Why is that echoing? I don't know. Somebody else was talking. Do you notice a difference between like that cell here and this cell here? Or between this cell here and, well, that cell's pretty good. So is that cell. You don't notice a difference between this cell here and that cell there? Okay, this cell is very distinct, very dark, very clear. The borders are very sharp. On that cell there, are the borders sharp? Come on, somebody answer.
No, they're not.
No, they're not distinct. They're not clear. The cell isn't clear. In fact, the cell almost looks ghostly, like a ghost. It's not really sharp. And that's probably that this cell is dying, and that cell is brand new and young and vibrant, okay? And oftentimes that's what you see when you see a pattern like this, and that is the cells that aren't doing well, they stain gram-negative like, and then the cells that are doing much better, they're staying gram-positive. And so we would call this a gram-positive cell, okay, because that's a dead cell. And the Mycobacterium, if you stain them correctly under the gram stain, they do stain lightly, gram-positively, okay, but they do resist the stain, and that's why I said they do stain gram-positively, but only lightly. And that is, there's so much macolic acid in them that they don't take up the crystal violet well, because it's a water-soluble stain and it doesn't get through the macolic acid of the cell wall. Whenever I've seen students who had a slide that looked like this, I asked them, how old is your cells? And they would always tell me, oh, it's a week old or 2 weeks old. And I said, okay, how old are the cells you're supposed to stain for the gram stain? Does anybody know? Anyone remember? Come on, somebody's got to remember. How old should the cells be if you're going to stain them for the gram stain?
24 hours.
That's close. You want them to be newly grown cells. So 24 hours would be perfect. 48 would be the maximum. Anything beyond 48 hours the cells may not stain appropriately, and that's usually what happened when students were staining their cells and they were staining like this. I found out that the cells weren't freshly grown, okay? They were at least a week old. So that's a problem with the gram stain. You have to have freshly grown cells, or the stain may not stain correctly. So in a sense, it is staining correctly. All of the new cells are staining gram positive, and then all of the old cells or dying cells are staining gram negative. Some tips in helping your cell stain well. Avoid using old cells. Prepare a very thin smear, ideally only one cell thick. Heat the bacteria on the slide after it's air dried. If you do it before it's air dried, the water in the cells will boil out of the cell, and that'll destroy the cell and destroy the shape of the cell. Another way of destroying the cells is if you overheat them and heat fixing. If you look at your cells and they look like they've melted, and they melt, instead of being an obvious bacilli, they'll start melting and there'll be a coccobacilli, and they'll be misshaped, and that's from overheating. Okay, obviously use fresh reagents. Don't over decolorize. If you over decolorize, you will get the and the gram positive, the crystal violet will be washed out. Usually though, one part of the slide will be staining gram negative and one part will be staining gram positive, and that's from over decolorization or maybe under decolorization. All right, any question about that? I'll have you read about the acid-fast stain. That's a differential stain for looking at bacteria that retain an acid stain, and that would be mainly the Mycobacterium. And you'll go through an in-class activity working with the acid-fast stain. And you would look at that like if you're testing a patient for Mycobacterium pneumoniae. So here's a acid-fast stain where the acid-fast negative stains stain greenish or bluish in this slide, and those would be normal bacteria. The acid-fast cells are the ones staining pink, and that would be a Mycobacterium species. There are special stains, such as a stain to stain for the glycocalyx or the cell capsule, and in this one we're using a negative stain. This would be something like cells that have a capsule, how the colonies look, and they'll look very mucoid on the petri dish because the capsule makes them very mucus. And this is a negative stain where you're staining the background. This is probably also a differential stain because it's staining more than just the background. It's also staining the cells. But you can see the cell here. It's very dark staining. That's why I'm saying it's probably a differential stain, that one cell stain is staining the cells, and the other stain is staining the background of the slide, lighter than the cells. And then this white region that isn't staining, that's from a capsule around the cell. Any question about that? So that would be Klebsiella pneumoniae, and the India ink is probably staining the background. But there's another stain in there because India ink doesn't stain the cells that dark. Another stain you can do is an endospore stain, and that's to test for different endospores. This table is showing you some of the species that have endospores and some of the clinical significance, like Bacillus anthracis causes anthrax, Bacillus cereus causes food poisoning, Clostridium tetani, you've probably heard of that one, causes tetanus, Clostridium botulinum causes food poisoning or botulism. And you'll notice that most of the bacteria that grow endospores are in mostly two families. There is an exception, but mostly members of the Bacillus family and members of the Clostridium family are the species of bacteria that produce endospores. And remember that an endospore is not a reproductive structure. It's a structure to allow these cells to survive when the environment becomes unfavorable for the parent cell to survive. And endospores are very hardy, and they survive all sorts of environments that normal cells would not normally survive. Let me see if I can blow this up. So in this endospore stain, well, that's a very good one right here. You can see the parent cell is reddish and the endospore is growing in between the parent cell. This one here has the endospore on the tip of that parent cell, and that endospore is very big. And there's an endospore that's been released from the parent cell. Anyways, this endospore, the endospore stains green, the parent cell stains pinkish. Any questions about that? There's also a Flagella stain. That's a special stain to stain flagella. Normally under the microscope, when you look at the cells, you will not see flagella because it's too thin of a structure. But if you use a special stain, it usually coats the flagella. Then you can see the flagella, and that we call the Flagella stain. This picture is showing you the different types of flagella. You don't need to know this for a test, but you do need to know it for the worksheet. It'll ask you a question about, I don't know, the different types of names for the different flagella. Like I said, you don't need to know it for a quiz, but you do need to know it for the worksheet. And it's just showing you that they have different names for where the flagella is found. Like if there's only one flagella, it's called monotrichous. And I'm just pointing this out because you have to answer it for the worksheet. And there's a Flagella stain showing you showing you flagella on at least 3 cells, maybe 4 cells. And these cells have many flagella on them. So they would be Petritrichus. Oh, excuse me, Peri, not Petri. Peritrichus. Which it says there. All right, so learn the terms. I'll let you read over that. And then go through laboratory exercise 4. Do procedure 1. Click on the link preparing bacterial smears and heat fixing. Know the procedure. We'll hold you to that. And then know the procedure for a simple staining. There's a video you can watch on making a simple staining. And simple staining, all the cells will look the same, so they'll all stain purple. You can tell difference, like between E. coli and Staphylococcus aureus, if the cells have a different shape, as they do here. And then work on the worksheet on procedure 3, it's going through a video on making the gram stain, and then you'll be doing a virtual lab on doing the gram stain. First, flood the slide with crystal violet, let it soak for 1 minute. Rinse the slide with distilled water, then flood the slide, we'll dump off the water, then rinse the slide with iodine, let it soak for 1 minute, and then rinse the slide with distilled water. Remove the water and then flood the smear with alcohol acetone decolorizer only for about 5 seconds. Like I said, it's best to watch while you're adding the decolorizer, and when the color stops draining from the slide, stop adding the decolorizer, and then immediately rinse with distilled water. And this is important that it be done immediately here. This one, it's not important that it be done immediately. You just need to do it before the dye dries. But here it needs to be immediate. And then fourth, flood the smear with saffron. Let it soak for 1 minute to 2 minutes. Rinse off the dye with distilled water. Pull off the water. Blot dry with bibulous paper, which hopefully you'll see that in the video. And then you can look at it under the microscope. This is showing you the different preparation. Initially, when you add grams crystal violet, all the cells will stain purple. When you add iodine, iodine only acts as a mordant, complexing with the crystal violet. There will be a little bit darker purple color. And then you decolorize. The gram-negative cells will be clear. The gram-positive cells will be purple if the gram stain has been done correctly. And then color with saffron. The gram-negative cells will stain pink. The gram-positive stains will be purple. Okay, here's 2 cells. What E. coli looks like under a gram stain, and what Staphylococcus aureus looks like under a gram stain. And then you're asked to determine the color of these, and you'll have to look up whether they're gram-positive, gram-negative, and I think that's in someplace in the lab. And then go through procedure 4, acid bacteria stain. Watch the video. I'm just going to say read through this. We'll be using carbol fuchsin as the stain. The first stain, the counter stain, will be brilliant green or methylene blue, depending on the stain used. And then the acid fast stain will stain reddish or pinkish, and then the acid negative stains will be greenish or blue. The acid fast stains will be the mycobacterium. And then special stains, there's videos on an endospore stain. And here you can see green staining endospores, the red staining, oops, that didn't work. I was trying to blow that up. The red staining perianthal cells. Okay. A special stain for flagella, you can see the flagella here. And then a special stain for the capsule. These cells have a capsule around the cell. And then the background. There are, I don't know, the background isn't uniformly colored. There are, what do you call it, bubbles or defects in the background. But you can clearly see the capsule from the defect in the background. All right, any question on the special stains? Then do the worksheet. Remember to get the link from the lab module for the worksheet, and then submit the worksheet. Are there any questions? If not, I will stay in the lab to answer any questions until 3:45. So you have about a half an hour to work on the lab, and I will be here if you have questions. If not, I'll see you next time.